If you want to assess tree risk at a professional level, you cannot stop at what you see on the outside. You have to understand what is happening inside the wood. Tree risk, as defined by ANSI A300 Part 9, is the combination of the likelihood of failure, the likelihood of impact, and the consequences of failure. The ISA Tree Risk Assessment Qualification Framework reinforces this structure. That first component, likelihood of failure, is where tree biology and wood structure matter most. Wood failure is not random. It is the result of decomposition, fiber disruption, and load exceeding structural capacity. So let's build this from the inside out. Wood is primarily composed of secondary xylem. We divide it into sapwood and heartwood. Sapwood is the outer functional xylem that conducts water. Heartwood is the inner non-conductive wood, often infused with extractives that can influence decay resistance. Decay patterns differ between these tissues. Sapwood rot affects the outer functional wood. Heartwood rot progresses within the central column. Understanding which tissue is compromised affects how we interpret structural stability. Wood strength is highly directional. Fibers run longitudinally. That alignment provides tensile and compressive strength along the grain. When decay fungi disrupt those fibers, strength decreases dramatically, often before major weight loss occurs. Wood decay is a biological process of decomposition driven by decay fungi. These organisms may act as pathogens, attacking living tissue, or as saprophytes, colonizing dead tissue. Some fungi begin as pathogens and transition into saprophytic behavior once tissue dies. In early stages, we may see incipient decay. At this point, discoloration may be present, but structural strength loss is not yet severe. Discoloration alone is considered a potential indicator, not a definite indicator of structural compromise. As decay progresses into intermediate decay, cell wall degradation becomes more extensive. Eventually, we reach advanced decay, where fiber integrity is severely reduced and residual wall strength may be critically compromised. Decay does not move randomly through wood. The compartmentalization of decay in trees model explains how trees attempt to limit spread. Wall 1 resists vertical movement. Wall 2 resists inward movement toward the pith. Wall 3 resists lateral movement across growth rings. Wall 4 is the barrier zone formed after injury. The barrier zone is chemically distinct from previous wood. It often contains Suberin and other defensive compounds. Suberin is a waxy substance that limits pathogen movement. Inside the tree, the reaction zone forms an existing wood to resist decay spread. Together, these defense systems influence the shape and stability of a column of decay. A column of decay is a vertical region of decomposed wood. Its size, symmetry, and interaction with structural defects influence likelihood of failure. Now let's talk about types of decay. Brown rot fungi preferentially digest cellulose and hemicellulose. They leave modified lignin behind. Wood often appears brown and cubical. Brown rot causes rapid loss of bending strength early in the decay process. White rot fungi degrade lignin as well as cellulose. Wood may appear stringy or fibrous. Strength loss patterns differ, but structural compromise is still significant. Soft rot often occurs in wetter environments. It progresses more slowly, but can still reduce structural capacity over time. We also categorize decay by location. Basal rot affects the lower trunk near the root flare. It often interacts with root decay. Basal swelling may be visible externally. That swelling can indicate internal dysfunction or compensation growth. Root rot compromises anchorage. When anchorage is reduced, the likelihood of whole tree failure increases even if the stem appears sound. Sapwood rot affects outer conductive tissue. Heartwood rot affects inner structural wood. Canker rot involves localized infections that kill cambial tissue, often creating sunken or dead areas. These can expand and disrupt load paths. Externally, we often detect fungal fruiting structures. These include conks and brackets. A bracket is a shelf-like fungal fruiting structure. A conk is a general term for a woody fruiting body. The presence of a fungal fruiting structure is a definite indicator of decay, but it does not tell us the full extent of the column of decay inside. Rhizomorphs are root-like fungal structures produced by certain species. Their presence at the base of a tree may indicate active root decay. That changes how we interpret likelihood of failure, especially under wind loading. Cavities are voids where wood has decomposed and been removed. A cavity alone does not define risk. What matters is residual wall thickness and load distribution. The outer shell carries most bending stress. If sufficient sound wood remains, structural capacity may still be adequate. Woundwood forms around injuries. It grows from the cambium to close wounds. Woundwood is structurally different from original wood. Its fiber orientation may alter strength patterns, but it represents the tree's attempt to compartmentalize and restore continuity. Discoloration within wood is often one of the earliest signs of dysfunction, But discoloration without significant fiber degradation is not necessarily structural failure. That is why it is categorized as a potential indicator rather than a definite indicator. A definite indicator would include advanced decay, extensive cavities, visible cracks through residual walls, or the presence of fungal fruiting structures associated with known aggressive decay fungi. When conducting a tree risk assessment under ANSI A300 Part 9, we are evaluating how these biological factors influence likelihood of failure. If basal rot has reduced cross-sectional area at the root flare, the bending moment from wind may exceed residual strength. If a column of decay intersects a crack, shear resistance decreases. If sapwood rot has progressed into structural latewood bands, modulus of rupture declines. We also consider species-specific compartmentalization ability. Some trees form strong barrier zones and robust reaction zones. Others allow decay to spread more freely. That affects how quickly incipient decay transitions into advanced decay. In the field, you should look for brackets, conks, rhizomorphs, cavities, cracks, basal swelling, and canker rot. You should evaluate woundwood formation and assess whether it is effectively restoring structural continuity. You should consider whether discoloration represents early dysfunction or deeper structural compromise. And always remember, decay fungi are part of natural ecological processes. Decomposition recycles nutrients. Not every decayed tree is high risk. Risk requires a target. A 300 Part 9 reminds us that risk is a function of likelihood of failure, likelihood of impact, and consequence of failure. A tree with advanced basal rot over a playground presents a different risk profile than the same tree in an unmanaged woodland. Common assessment errors include overreacting to a cavity without measuring residual wall thickness, underestimating root rot because the canopy appears healthy, and assuming that all conks indicate imminent failure. Tree risk assessment is the integration of wood biology, structural mechanics, and target evaluation. When you understand sapwood and heartwood dynamics, brown rot versus white rot behavior, the role of the barrier zone and reaction zone, and how a column of decay alters load paths, your assessments become more precise. Trees fail when mechanical stress exceeds structural capacity. Structural capacity is shaped by wood anatomy, decay progression, compartmentalization strength, and residual wall thickness. If you want to assess risk accurately, you must read the wood, not just the surface, but the biology within it. Thanks for listening to the Arborist podcast. Let's keep building your arborist knowledge one domain at a time. If you found this useful, please like and subscribe.